Today we are going to discuss the techniques which are used to detect the mutation. But before we move forward, I want to discuss about the meta-PCR and why it is essential. There are many genes which are having multiple exons. Like they have, suppose one gene has exon 1 till exon 24 or exon 60. If the single gene has many exons, and if you want to do Sanger sequences and exon by exon, this is going to be very labor intensive and also costly. So to reduce that, meta-PCR is basically used. So for example, if you see here, in this what happened, we are using one set of primers, let's say for exon 1, and the another set of primers for exon 2. So if you can see, each primer pair includes a spatial sequence 5' extension that is not to the next exon amplicon. So if you see here, this is the primer pair 1 and this is the complementary sequences TGCA. So what will happen? This particular TGCA will join here. This particular TGCA will join here and it will link the exon 1 and exon 2. So amplicons link together end to end, and that is known as the concatenation. And why it is done? Just to save time and just to save money. If your gene is having many exons and you don't want to do sequencing for every exon, then you can go ahead and do the meta PCR. So what are the heteroduplexes? Heteroduplexes means the is a is a is a DNA double strand formed by two similar but not identical sequences. Means one strand of DNA has the mutated sequence and the other strand of DNA has the normal sequence. So what will happen if you see here? So let's say if this is normal sequence, that will be having ACC and the mutated sequence will having AGC. Let's say. So there is a mutated sequence, and let's say this is just an example. This is the normal sequence. So when they will mix together, they will form the heteroduplex. Definitely they are going to form the homoduplex also, because what happened, this is, let's say, your normal DNA, this is your mutated DNA, this is normal, this is mutated, both will separate. Normal will separate, mutated will separate. Let's say this is not, this is normal, this is normal, this is mutated, this is mutated. So when you will combine them, normal can reannul with n, normal can reannul. Let's say this is 1, this is 2, this is 3, and this is 4, then 1 can join with 1. There will be many combinations. So this is homoduplex. 1 can join with, 1 can join with 2, that is homoduplex. 1 can join with 3, that is heteroduplex. 1 can join with 4, that is heteroduplex. 3 can join with 4, that is again homoduplexes. So these are the examples of heteroduplexes. Heteroduplexes means when one strand has normal DNA and the other strand has the mutated DNA. For example, 1 and 3, 1 and 4, or you can say 2 and 3, 2 and 4. These are the heteroduplexes. And how you can identify them? You can identify them by the denaturing gradient gel electrophoresis or denaturing high performance liquid chromatography. So what happens in the denaturing gel gradient gel electrophoresis? So what happens in this, what you will do, you will design the primer pairs that will against the normal DNA, and you will also design the primer pair that will against the mutant DNA. Okay. And each of these primer pairs will have the GC clamp at the 5' end. Mutated DNA also will, primers that is designed for the mutated DNA will also have the GC clamp. And why GC clamp? Because G and C, they have the triple bond, triple hydrogen bond. So they are highly stable. So what happened? It holds parts of the DNA tightly. So now if you will see here, special primer, one of which includes the GC clamp that holds parts of the DNA tightly. Now you will run the gels in a special gel. Gel will contain the denaturants, so it will denature your DNA. And what could be the denaturants like urea, formamide, from low to high? As DNA moves through the gel, it starts to melt at specific points depending on its sequence. So homoduplexes and heteroduplexes will run at a different speed, and because of that, you can easily identify them. If suppose you are having the primer pairs for the normal DNA and the primer pairs for the mutated DNA, and once they are denaturing, as I explained in my previous slide, they can reanneal with each other. They can also reanneal with the mutant DNA. We are specifically interested in when the normal DNA is interacting with the or reannealing with the mutant DNA, because in that case it will form the heteroduplex, and once it is forming the heteroduplex, its velocity is going to be changed, its denaturation is going to be changed depending on its sequence. So you will find the normal and the mutant DNA at the different positions. For example, this is a clear example where you can see these are the two patient samples of the cystic fibrosis, and the PCR was done for chromosome exon number 6, 17, 24, and multiple exons. You can see the patient number 1 that is forming the homo as well as the heteroduplex, because homoduplex are running faster. But in case of the P2, it is absent. Similarly, in case of the patient P2, you can see there are the variants for the heteroduplexes. This is the homoduplex as like the patient P1, but this is the heteroduplex like because of the mutated sequence. And in the 17 exon, similarly for the 24th exon also you can see in the patient number P2, it is having the variant of chromosome gene exon number 24. So that is how you can easily identify that this patient is having what mutation and this patient is having what mutation. But there are some limitations of denaturing gradient and electrophoresis, because in this you have to do the PCR, and PCR is optimal for 150 to 500 base pair products, and as you increase the number of fragments, it reduces the resolution. It can identify the mutation, whether it is present or absent, but it cannot find out the exact location like 1295, 123. What is the position? It cannot identify. You have to use a GC clamp, and it requires a careful attention. How many GC you are going to add? It requires validation also. Sometimes the silent, like the synonymous mutations, they might not cause the detectable mobility shift. It is a labor intensive technique. It is low throughput because you have to do PCR, then you have to do page. In both of these techniques, if you are running for 1,000 samples, it is going to be taking too much of time. So it is labor intensive and low throughput. Not fully quantitative, it can identify whether the person is heterozygous or the homozygous. For example, in this case, you can say the person is heterozygous for this particular exon 17. In this case, you can say the patient P1 is heterozygous for exon 6. But you cannot identify quantitatively, okay, like the real-time PCR. And it requires the wild-type controls are necessary to identify the abnormal patterns. And why it is so? Because if suppose you have only wild-type sequence or mutated sequence, they will not form heteroduplexes. So how you will identify? So in that case, you have to add the wild-type DNA. and high GC content may form stable structures that sometimes hinder the denaturation. Now, coming to the second technology which is used to identify the mutation is the chemical cleavage of the mismatch. What they will do once there is a heteroduplex DNA is formed, that is forming the kings or bubble. For example, here you can see they can form a bubble-like structure at the position of heteroduplex. And this bubble-like structure is recognized by the chemicals like hydroxylamine or sodium tetroxide. They will cleave this product. If there is a mismatch or there is a heteroduplex formation, there is going to be the cleavage, and that you can identify easily. For example, this person did the meta PCR that is having the 1032 base pair product. This is the control part. This is the control sample. But in case of the mutant sample, you are getting the homoduplex region, this particular product. But this, because if this person is forming the heteroduplexes as well, so hydroxylamine will cleave this 1032 product to the 813 base pair and the 239 base pair, and because of that you can see the different peaks and you can identify that this person is going to have the mutated sequence. Always remember, as I explained in last my slide, whenever there is a denaturation and prenaturation, if suppose this is your normal DNA and this is denatured and this is your mutated DNA, this is also denatured, always remember there are chances that normal can reannihilate with normal will form the homoduplex. There are chances, right? Normal can reannihilate with mutant sequence and it will form the heteroduplexes like happened in this case. As you can see here, it is also forming the homoduplex, but it is also forming the heteroduplexes as well because there is a presence of mutation. So you can say that this person is having the mutated sequence. Now coming to the high performance liquid chromatography, again because if there is a mismatch, there is a heteroduplex formation, they will migrate at a different speed in a special chromatography column, and this will create a separate peak. For example, this is the DMD mutation because DMD mutation is X-linked, and because males have only one X chromosome, so you have to add the wild type sequence. Otherwise, what will happen, because this X chromosome is mutated, it will only form the homozygous condition. So you have to add the wild type sequence to form it, the heteroduplex. Okay. So for example, here you can see this is the control sample and this is the mutated sample because there is a heteroduplex pair which you can see here. This particular region is the heteroduplex. And this is, you can see, the homoduplex. Now, another technology is single-stranded conformation polymorphism. In this, you amplify the DNA and then you denature the DNA. And then you snap hole immediately. So rapid cooling prevents re-annealing and each strand folds into the unique shape based on its sequence. Now the sample will run not just because of their size but because of their shape as well. For example, if you see here, the single-stranded DNA will run more slowly and you will see that in the upper part, and the double-stranded DNA will run more faster. And because it is forming the heteroduplex, you can see the patient number 3 and patient number 7 is forming the heteroduplexes because you are getting the heteroduplex formation, because this is the homoduplex and you are getting the heteroduplex portion as well. So you can identify that patient number 3 and 7 is different than the other patients. And there are some limitations as well. Limited detection of large mutations and repetitive regions. There is a sensitivity issue because this is done by autoradiography, particularly for, and in this also you can't rely on these kind of results if you are doing the patient samples, because there might be some mis loading or something. You add something by by chance, and you can also get a band because of the contamination. So it is not a very reliable technique. Requires the optimal condition, complexity in interpreting patterns and multiple conformation. That's a very important point. Labor intensive and not suitable for high throughput screening. Now coming to the protein truncation test, what happened in this case? Suppose you have a protein that is 140 kD, but somehow there is a mutation and you are getting the protein of 70 kD. Why it is so? Because this is your protein, and because of suppose there is some frame shift mutations, you will get the stop codon UAA, UAG or UGA at 70 kD position, and that is why your protein is truncated. And if this is truncated, this will migrate very faster in the STS page. So you can see, this is the thing which you can see. This protein is truncated, this protein is truncated. So you can easily identify because the protein size is this much. But here you are getting a very small size protein because the stop codons come there. So this is the protein truncation testing. It is limited to the premature stop codons, and it missed the other types of mutations. Okay. False negative for mutations not leading to significant truncation or due to outside coding region. Dependency on the expression system for reliable detection of truncated proteins. Difficulty in detecting alternative forms of the truncated proteins. RNA and cDNA quality are very essential. If RNA is degraded, leading to the possible false negatives. Complexity in interpretation of gel patterns, especially with overlapping or non-specific bands. Not suitable for high throughput screening or large scale genomic studies, and it does not provide functional insights into the truncated proteins. It will not identify if it is having the carbohydrate, it's a glycosylated protein, so what kind of protein it is. You cannot identify them. So that's all about today's lecture.